Welcome back to Dr. Liu's VCE Biology. Today's topic is responding to antigens and acquiring immunity, as presented in the 2025 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. Why option B is correct, the category. In the VCE study design, the first line of defense is defined as the innate, non-specific barriers that exist to prevent infection. Because plants do not have a circulatory system to transport specialized immune cells, the second line, their chemical defenses, like these oils and phenols, are essentially the front line of their survival. They are often pre-formed, constitutive, and present in the cells before a pathogen even arrives, making them a literal line of defense. Why option A is correct, the mechanism. Option A describes the mode of action. Essential oils and phenolic compounds are biologically classified as antimicrobial agents. Their chemical structure allows them to disrupt the cell membranes of bacteria or inhibit the enzymes of fungi. This provides the functional how to the what of option B. C is incorrect because microbiota refers to the community of microorganisms living in a specific environment. It is not a classification for a chemical compound. D is incorrect because essential oils and phenolics are chemical or biochemical defenses, not structural, like a waxy cuticle or cell wall. Correct answer, D, Student 4. In scientific inquiry, an anecdote is a personal account or individual story used as evidence, which lacks the rigor of a controlled study or statistical analysis. Student 1 is discussing a type of formal scientific investigation, correlational study. Student 2 is stating a hypothesis or opinion. Student 3 is referencing a scientific study, formal peer-reviewed evidence. Student 4 is sharing a personal story about her mother's experience. This is a classic example of anecdotal evidence, which may be true in that specific instance, but is not scientifically generalized. A is incorrect. A virus is non-cellular, and a fungus is cellular, the reverse of what is listed. Also, a prion is a non-cellular pathogen, not typically classified as a common allergen. B is correct. Bacterium, unicellular prokaryote, cellular. Virus, genetic material encased in protein, non-cellular. Dog hair, a common environmental trigger for an allergic response, allergen. C is incorrect. A bacterium is cellular, not non-cellular. A virus is a pathogen, not an allergen. D is incorrect. A prion is non-cellular, and dog hair is an allergen, not a pathogen. In VCE biology, distinguishing between the structure of pathogens and the nature of antigens is a fundamental skill. Cellular pathogen, organisms made of one or more cells, prokaryotic or eukaryotic, that can reproduce independently, such as bacterium, fungus, protozoa, helminths or worms. Non-cellular pathogen, infectious agents that lack a cellular structure and require a host cell to replicate, or are simply misfolded proteins, such as virus, prion. Allergen, a non-pathogenic substance, often a protein, that triggers an overreaction of the immune system, Ig-mediated, such as dog hair, pollen, dust mites, peanuts. This question tests your understanding of the role of the secondary lymphoid organs, specifically lymph nodes, in the initiation of the adaptive immune response. Why A is incorrect. Swelling in lymph nodes is due to the proliferation of white blood cells, lymphocytes, and increased lymph fluid, not an accumulation of red blood cells. Red blood cells stay within the circulatory system unless there is a hemorrhage. YB is correct. Lymph nodes are the primary site where antigen-presenting cells, APCs, such as dendritic cells, meet naive T and B lymphocytes. When a lymphocyte with a receptor specific to the pathogen's antigen is found, clonal selection, it begins to rapidly divide, clonal expansion. The massive increase in the number of these specific lymphocytes is what causes the lymph node to physically swell. YC is incorrect. Both macrophages and dendritic cells are antigen-presenting cells. In the lymph nodes, it is the dendritic cells, or macrophages, that present antigens to T helper cells, not macrophages presenting to dendritic cells. YD is incorrect. While lymph fluid is filtered in the nodes to trap pathogens and debris, lymphocytes are not removed from the system there. Instead, the lymph nodes serve as a hub where they congregate to screen for antigens before returning to the bloodstream via the thoracic duct. The correct answer for question 26 is D. Blood flow is increased to the infection site, allowing more immune cells to reach the area. This question covers the inflammatory response, which is a vital component of the second line of defense in the VCE biology curriculum. YD is correct. During inflammation, damaged cells and mast cells release signaling molecules like histamine. Histamine causes vasodilation, widening of blood vessels, and increases capillary permeability. This results in increased blood flow to the area, which brings more leukocytes, such as neutrophils and macrophages, to the site of infection to combat pathogens. YA is incorrect. Natural killer, NK, cells do not engulf pathogens. They destroy infected or abnormal self cells by releasing cytotoxic chemicals, like perforins and granzymes, that induce apoptosis. Phagocytes, such as macrophages and neutrophils, are the ones that engulf pathogens via phagocytosis. YB is incorrect. In an inflammatory response, the number of white blood cells at the infection site increases, not reduces. The body often ramps up production and recruitment of these cells to manage the threat. YC is incorrect. The inflammatory response is actually an activation of the innate immune system. While chronic inflammation can cause internal damage, the primary purpose of the response is to eliminate the source of cell injury and initiate repair. Triple strike logic for students. When teaching the why behind the symptoms of inflammation: redness, swelling, heat, pain. Redness, heat, caused by increased blood flow, vasodilation. Swelling, caused by increased permeability, allowing fluid and immune cells to leak into the tissues. Purpose: to localize the infection and accelerate the arrival of phagocytes. YA is incorrect. Histamine is a signaling molecule, an organic nitrogenous compound, not an enzyme. While it does cause vasodilation, its chemical classification in the characteristic column is wrong. YB is incorrect. Interferons are indeed produced by virally infected cells, but their function is to signal neighboring cells to heighten their antiviral defenses, e.g., by producing enzymes that degrade viral RNA. They do not break down bacterial cell walls, that is the job of lysozymes. YC is correct. Complement proteins are a group of about 30 proteins that circulate in the blood in an inactive state. When they encounter a pathogen, they trigger a complement cascade that leads to opsonization, tagging pathogens, chemoattraction, recruiting phagocytes, and the formation of a membrane attack complex MAC, which causes cell lysis bursting of the pathogen. YD is incorrect. Lysozymes are enzymes found in secretions like tears and saliva that digest bacterial cell walls. They are not primarily produced by mast cells, which produce histamine, and they do not act as signaling molecules for adjacent cells. Why A is correct. Plasma cells are the differentiated progeny of activated B lymphocytes. Their sole, primary biological role is to act as antibody factories. They secrete vast quantities of soluble proteins, antibodies and immunoglobulins, that travel through the blood and lymph to neutralize pathogens. Why B is incorrect. Plasma cells do not perform phagocytosis. Furthermore, destroying virally infected cells is the role of cytotoxic T cells via apoptosis, or natural killer cells, not phagocytosis. Why C is incorrect. While many immune cells secrete cytokines for communication, this is the primary defining function of helper T cells, not plasma cells. Why D is incorrect. Antigen presentation to helper T cells is performed by antigen-presenting cells, APCs, like dendritic cells, macrophages, and naive B cells. Once a B cell has become a plasma cell, it has already passed the presentation stage and is now an effector cell. State two roles of neutrophils in an immune response to meningitidis. Phagocytosis. Neutrophils engulf and digest the bacterial pathogen using lysosomal enzymes. Secretion of cytokines. Neutrophils release cytokines to recruit other immune cells to the site of infection, enhancing the inflammatory response. Contrast how the adaptive immune system responds to an extracellular pathogen, like meningitidis, and an intracellular pathogen, like the influenza virus. Extracellular response. Humoral extracellular pathogens, like meningitidis, primarily trigger the humoral immune response. B lymphocytes differentiate into plasma cells, which secrete antibodies specifically designed to neutralize the pathogen, or opsonize it for phagocytosis. Intracellular response. Cell-mediated intracellular pathogens, like influenza virus, primarily trigger the cell-mediated immune response. Cytotoxic T cells recognize viral antigens presented on MHC Class I markers of infected host cells and release perforin, agranzymes to induce apoptosis. Contrast point. The humoral response targets pathogens in bodily fluids, extracellular, whereas the cell-mediated response targets and destroys infected self cells to stop viral replication, intracellular. 1. The barrier and evidence check. Plant defense, chemical, phenolics of oils, equals preformed and non-specific. Note, plants lack a circulatory system of immune cells. Scientific rigor, anecdotes, personal stories, is not equal to evidence. Scientific studies must be replicable and peer-reviewed. 2. Pathogen and molecule classification. Pathogens, cellular, bacteria or fungi, versus non-cellular, viruses or prions. Allergens, non-pathogenic, trigger Ig-mediated responses. Key innate molecules. Histamine, signaling molecule, vasodilation, not an enzyme. Interferons, viral warning signals to neighboring cells. Complement, inactive blood proteins, cause cell lysis when activated. Lysozyme, enzyme, tears a saliva that digests bacterial walls. 3. The battlefield, lymph nodes and inflammation. Lymph nodes, site of clonal selection or expansion. Swelling equals increase in lymphocytes, adaptive response, not RBCs. Inflammation, mast cells release histamine, induce vasodilation, and increase permeability, induce redness, the heat or swelling. To master the adaptive immune system, You must distinguish between two specialized strategies based on where the pathogen is located. First, we have the humoral response, which targets extracellular pathogens, those floating freely in your bodily fluids, like the bacterium Neisseria meningitidis. The star of this response is the plasma cell. Once activated, these cells act as factories, secreting antibodies, or immunoglobulins, into the blood. These antibodies work through neutralization and opsonization, essentially tagging the bacteria so phagocytes can find and destroy them more easily. However, if a pathogen like the influenza virus slips inside your cells, antibodies can no longer reach it. This triggers the cell-mediated response. Here, the effector cells are cytotoxic T cells. These cellular assassins don't look for free antigens, instead, they recognize viral fragments presented on a cell's MHC Class I markers. Once a target is identified, the T cell releases perforin and granzymes, forcing the infected host cell to undergo apoptosis, or programmed cell death. In short, the humoral response uses antibodies to clean up the fluids, while the cell-mediated response uses T cells to eliminate the infected factories from within. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.